Hey everyone, welcome back to the channel, your destination for exploring the deep, mysterious corridors of the human mind, where psychology meets science and ancient wisdom finds its reflection in modern discovery. Today, we're about to take a journey that will stretch your imagination, ignite your curiosity, and maybe, just maybe, shift the way you see your own inner world. We're diving into something absolutely mind-blowing, a discovery so small, so subtle, you might miss it if you blink. And yet, it has the power to rewire how our brains function. It's a tiny genetic switch called mini exon B, a microscopic fragment hidden inside our DNA. But don't be fooled by its size. This little switch plays a massive role in controlling the balance between thought, emotion, and behavior. It's like finding a pebble that tips an entire mountain. And here's where it gets really fascinating. We're not just going to look at this discovery through a scientific lens. We're also going to explore it through the eyes of Carl Jung, one of the most profound psychological thinkers of the 20th century. Jung believed our minds weren't just shaped by personal experiences, but also by ancient universal patterns he called archetypes. He proposed that deep inside all of us lies a blueprint, a kind of inherited memory, shared across all cultures, all times. So here's the question we're going to explore today. Could this tiny genetic key be part of the code that connects us to those ancient archetypes? Could the science of genes and the soul of psychology be pointing to the same mysterious truth? Stick around, because what we're about to uncover is not just fascinating science, it's a window into the very nature of who we are. Science meets psychology. This genetic switch, mini exon B, is almost invisible to the naked eye, just a few nucleotides long. But what it does is extraordinary. Imagine your mind as a grand symphony with thousands of instruments playing together in harmony. Mini exon B is like a single note that, when struck, changes the entire rhythm. It tunes the tension between chaos and calm, between thought and silence. And when we bring Carl Jung into the picture, it's like putting a mythological filter over a microscope. Jung's concept of the psyche, divided into conscious awareness, personal unconscious, and the collective unconscious, maps onto the brain in ways we're only beginning to understand. Jung believed that symbols, dreams, and behavior all emerge from deep psychic patterns formed through both biology and collective history. So when science shows us that a tiny bit of DNA can shift behavior, emotion, and social interaction, it raises a profound question. Is our biology hardwired not just for survival, but for story, myth, and meaning? Whether you're a neuroscience nerd, a psychology buff, or just a curious soul wondering why you think, feel, or dream the way you do, this journey is for you. Breaking down the science. Let's begin with the basics. Recently, researchers identified a tiny snippet of genetic code, mini exon B, within a gene called PTP delta. Now, don't let the complex names intimidate you. Imagine your DNA as an enormous instruction manual, the kind that would take lifetimes to read. Each gene is a chapter in that book, and each exon is a paragraph providing very specific directions. Mini exon B, that's one of the tiniest paragraphs, just a few lines. And yet, it determines how parts of your brain wire together. It's like a sentence in a recipe that changes how you bake the entire cake. Leave it out, and everything comes out flat or bitter. Include it, and you get balance, complexity, flavor, something whole. The role of PTP delta and neuron balance. The PTP delta gene plays a critical role in shaping how neurons, the cells in your brain, connect with each other. These connections, called synapses, are like circuits in an incredibly sophisticated supercomputer. And as in any system, balance is key. Mini exon B acts like a micromanager for this wiring process. It influences two vital types of neurons, granule cells, which send excitatory signals and ramp up brain activity, and interneurons, which inhibit activity and calm things down. Imagine your brain as a city. Granule cells are like streetlights turning green, saying go, go, go. Interneurons are red lights and stop signs, keeping the city from descending into chaos. Now picture what happens when those lights are out of sync. Too much green light, and your thoughts race uncontrollably. Too many red lights, and your motivation shuts down. This is the balance that mini exon B helps maintain. When the switch fails, here's where the story takes a dramatic turn. In mouse studies, when scientists removed mini-exon B, the entire balance crumbled. Granule cells weakened, interneurons became overly dominant. The neural symphony lost its rhythm. The mice exhibited behaviors that were hauntingly familiar: anxiety, hypervigilance, decreased movement, and lower survival rates. It was as if removing that one sentence from the brain's instruction manual made the entire story collapse. And this isn't just about mice. In humans, disruptions in the same genetic pathway are linked to serious neurodevelopmental conditions, like autism spectrum disorder, ADHD, and anxiety disorders. One tiny exon, one critical switch, may be central to how the brain learns to balance itself between stimulation and rest, between presence and withdrawal, between order and disarray. This isn't just science, it's a glimpse into the machinery behind our moods, thoughts, behaviors, and maybe even our dreams. Alternative splicing and the brain's final cut. To understand how mini-exon B does all this, we need to talk about something called alternative splicing. Think of a gene as raw footage from a film. Splicing is the editing process. It decides which scenes make the final movie. Mini-exon B is like a crucial scene that gives the whole story emotional depth. Cut it out, and the film loses coherence. The characters lose purpose. The plot twists don't land. That's exactly what happens in the brain. Without mini-exon B, the edited movie of the brain, the neural connections, behavioral responses, and emotional balance loses its narrative arc. It becomes fragmented, chaotic, and the person or the mouse suffers. This shows us just how incredibly precise our bodies are, how evolution has fine-tuned us, not just for physical survival, but for emotional coherence, for balance, for story. Carl Jung, the bridge between science and soul. Now let's step into the realm of depth psychology, where the science of neurons meets the soul of myth. Carl Gustav Jung was a Swiss psychiatrist who believed that our psychological makeup wasn't just personal, it was universal. Beneath our everyday thoughts lies what he called the collective unconscious, a vast reservoir of inherited images, instincts, and symbolic patterns shared across humanity. To Jung, this wasn't just theory, it was the root of our art, our religions, our dreams, our fears. He believed that these ancient patterns, these archetypes, are etched into the very fabric of our being. So what happens when neuroscience starts finding these etchings in our very DNA? What if something like mini-exon B is part of the biological scaffold that shapes not only how we think, but what we think about? Could the collective unconscious have a physical basis, built into the silent language of our genes? Maybe mini-exon B isn't just a switch for neuron balance. Maybe it's part of the key that opens the doorway to the archetypal mind. Picture the collective unconscious like an ocean we all share. It's filled with archetypes, universal patterns or blueprints for how humans think, feel, and behave. These archetypes show up in myths, dreams, and stories across cultures, like the hero, the wise old man, or the shadow, which represents our darker side. Jung believed these patterns come from our evolutionary past, hardwired into our brains through generations of human experience. Jung thought these archetypes live in the older, deeper parts of our brain, the subcortical regions, like the amygdala or hippocampus, which handle emotions and instincts. This is where our genetic switch comes in. Mini-exon B affects how neurons connect in these ancient brain areas. So in a way, it's shaping the biological foundation of what Jung called the collective unconscious. Jung also believed the psyche is self-regulating. It's always trying to balance opposites, like conscious and unconscious, or excitement and calm. When that balance is off, you get psychological problems, like anxiety or dissociation. Sound familiar? The mini-exon B study showed that messing with this genetic switch disrupts the brain's excitation-inhibition balance, leading to behaviors that Jung might say reflect a disturbed psyche. It's like the brain's hardware is struggling to run the psyche's software. Another big idea from Jung is individuation. Another central pillar in Carl Jung's psychology is the concept of individuation, the lifelong journey of becoming who you truly are. It's not just about growing up or learning skills. It's a deep, often painful unfolding of the soul, a process where the conscious and unconscious parts of the psyche gradually integrate. It's the inward spiral toward authenticity. Jung believed that this journey begins even before we're aware of it, perhaps even before we are born. And here's where modern science intersects with timeless psychology. The discovery of mini-exon B suggests that the architecture of our psyche, the raw scaffolding for emotional and cognitive balance, may depend on genetic whispers encoded in our DNA from the very start. This tiny switch helps set the rhythm of neuronal connections that later orchestrate everything from perception to emotion. If something misfires in these earliest moments, if mini-exon B is absent or misread, it might not just alter brain chemistry, it might ripple across a lifetime, influencing how easily one can access or embody the archetypes necessary for wholeness. In Jungian terms, it could hinder individuation itself, making it harder for the self to become whole, for the unconscious and conscious to meet in harmony. So how do we tie this tiny genetic switch to Carl Jung's big ideas? So how does a fragment of code, so small you need an electron microscope to even begin to see it, tie into Jung's sweeping vision of the human soul? Let's connect the dots. Jung believed that deep beneath our conscious thoughts and feelings lies a vast reservoir he called the collective unconscious. This isn't just a poetic metaphor, it's a psychic blueprint, an inherited foundation built over millennia, filled with archetypes, universal images, and instinctual motifs that shape how we experience life. Now think about this. DNA is also a kind of inherited blueprint, passed down not just from parent to child, but from the entire evolutionary lineage of humanity. Mini-exon B is part of that lineage, a sliver of biological memory. It tells the brain how to wire itself in a way that supports essential functions: emotions, movement, self-regulation. Jung might have seen this not as a contradiction to his theories, but as their missing piece, a literal genetic basis for the psychic patterns he intuited. A key that suggests the collective unconscious may not just be metaphysical, it may also be molecular. First, let's talk about the collective unconscious. Let's return to the collective unconscious. Jung described it as a living archive, an inherited vault of ancestral experience stored deep within us. And modern neuroscience offers an intriguing parallel: epigenetic and genetic memory. The way DNA encodes survival strategies, emotional responses, and behavioral tendencies over generations. Mini-exon B is part of this coding, specifically influencing synaptic wiring in the cerebellum and cortex, areas involved in movement, emotion, and perception. These aren't just mundane brain functions, they are the stage upon which archetypes come alive. When Jung spoke of the mother archetype, for instance, he wasn't just talking about one's personal mother, he meant a deep universal imprint of nurturance, comfort, and protection. That archetype must find its neural echo somewhere in the brain. And what if mini-exon B is part of the script that builds the circuits for this echo? It's speculative, yes, but poetic speculation often precedes scientific discovery. If these circuits are improperly formed, we might see social disconnection, emotional numbness, or behavioral rigidity, traits seen in autism and ADHD. The pattern is there, but the connection, the energetic bridge to the archetype, is incomplete. Here's a cool thought. What if mini-exon B is part of the machinery that builds the neural circuits for archetypes? Imagine for a moment that mini-exon B acts like a silent engineer, laying down the basic circuits that will later host the living expression of archetypes. The hero, the shadow, the mother, the trickster, these timeless forces need a physical framework to express themselves. Without it, the music of the psyche can't play in tune. In disorders like ADHD or autism, what we often see isn't a broken person, but a person whose neural symphony is playing a different rhythm. Perhaps the conductor, the genetic script, is missing a crucial line. Jung believed archetypes were potentials, not guarantees. They need conditions to emerge properly. So what if this tiny genetic piece helps determine whether those conditions are met? Now let's zoom in on that excitation-inhibition balance. Now let's return to the core mechanism at play, the balance of excitation and inhibition in the brain. Without mini-exon B, this balance tilts. Some neurons become overactive, while others go quiet. It's like an orchestra where the violins scream while the cellos disappear into silence. The result? Anxiety, confusion, immobility, traits that mirror a psyche out of alignment. Jung would have seen this as a biological metaphor for psychic disharmony. He often emphasized the need to balance opposites, light and dark, masculine and feminine, ego and shadow. In a healthy psyche, these forces dance together. But when one dominates, the dance turns into a struggle. Without mini X on B, the brain can't maintain this essential balance, not just in movement or attention, but in the rhythms of the self. The micro becomes the macro. The molecular dissonance becomes emotional turbulence. Let's get a bit speculative, because that's what Jung was all about. Let's follow Jung into the realm of imaginative science, where metaphors breathe alongside molecules. When mini exon B was removed in mice, not only did they become anxious, they showed a decreased will to explore and even reduced chances of survival. What if this is more than behavior? What if it's a kind of spiritual withdrawal, a fragmentation of the ego's ability to contain the unconscious? Jung warned that when the unconscious overwhelms the ego, we fall into chaos. Our instincts run wild, or our emotions become unbearable. In extreme cases, the very will to live can vanish. This aligns eerily with what we see in these mice. With one tiny switch turned off, the entire system begins to collapse. The self, the higher order of unity Jung spoke of, is no longer supported by the infrastructure below. This balance is a big deal in Jung's world. To Jung, balance wasn't optional, it was sacred. He used the symbol of the yin and yang to represent the harmony of opposites. Day and night, creation and destruction, conscious and unconscious. Mini exon B could be seen as the biological equivalent of this symbol, a genetic mechanism that keeps the psyche's dualities in dialogue rather than conflict. When that switch fails, the result is fragmentation. And in that fragmentation, we see echoes of what Jung called the neurosis of the modern world. An inner split, a loss of wholeness. The mice with altered mini exon B were not just biologically compromised. They were, in a symbolic sense, embodiments of the unindividuated psyche, caught in imbalance, unable to integrate. So perhaps the lesson here is profound. The journey to the self doesn't begin in the therapist's office. It begins in the womb, in the first sparks of neuronal fire, in the invisible instructions written into our DNA. Jung's dream of integration may be more than a metaphor. It may be written in the code of life itself. And then there's individuation. Jung's idea that we spend our lives becoming whole by integrating all parts of ourselves. The mini exon B study shows that this genetic switch is active during early brain development, setting the stage for how our neural circuits form. If something goes wrong at this stage, it could affect the psyche's ability to integrate later in life. Jung might say that a disruption in mini exon B is like a roadblock on the path to individuation, making it harder for the psyche to find its balance. Okay, so why should you care about this? Whether you're into neuroscience, psychology, or just curious about what makes us human, this discovery is a big deal. It shows us how something as small as a genetic switch can shape who we are, our behaviors, our emotions, even our mental health. And when we bring in Carl Jung, it gets even more profound. His ideas give us a way to think about how our biology connects to the deeper, universal parts of being human. Let's be real, Jung's ideas, like the collective unconscious, aren't something we can stick under a microscope. They're philosophical, speculative, and sometimes a bit mystical. But that's what makes them so powerful. The mini exon B discovery gives us a concrete piece of the puzzle, how our genes build the brain's foundation. Jung's theories give us a way to dream bigger, to ask, could these genetic switches be the biological roots of the archetypes that shape our stories, our dreams, our lives? Think about it. Our ancestors didn't know about DNA, but they told stories about heroes, gods, and monsters. Stories that Jung said come from the collective unconscious. Today, we're finding that tiny genetic pieces, like mini exon B, might be part of how those ancient patterns got wired into our brains. It's like science is catching up to the myths, showing us that the stories we tell and the behaviors we show are rooted in our biology. This also matters for mental health. The study links mini exon B disruptions to conditions like autism and ADHD. Understanding these genetic mechanisms could lead to better treatments, maybe even ways to support brain balance early in life. And from a Jungian perspective, it's a reminder that mental health isn't just about fixing symptoms. Jung would probably say that discoveries like this are a call to action. They remind us to pay attention to our inner world, to work toward balance, and to embrace the journey of individuation. Whether it's through therapy, creativity, or just reflecting on our dreams, we can help our psyche find that equilibrium. Just like our brain needs the right genetic switches to function. Now let's hit pause and talk about the limits of this connection. Jung's ideas are fascinating, but they're not science in the way we think of it today. The collective unconscious and archetypes are hard to prove. They're more like frameworks for understanding the human experience. Some critics say Jung's theories are too mystical, too focused on symbols, and not enough on hard data. And there's a practical question. Can our genome really encode something as complex as archetypes? Back in the early 2000s, some scientists argued the human genome was too small to carry that kind of information. But recent research, like the mini exon B study, shows that tiny genetic elements can have massive effects through processes like alternative splicing. So maybe the genome is complex enough to shape the kinds of universal patterns Jung talked about. Still, we have to be careful. Linking a genetic switch to something as big as the collective unconscious is a stretch. It's more of a thought experiment than a proven fact. The mini exon B study is about synaptic connections and behavior, not directly about archetypes or the psyche. But that's the beauty of Jung. He invites us to think beyond the lab, to see connections between science and the soul. So what have we learned today? A tiny genetic switch, mini exon B, plays a huge role in balancing our brain's excitatory and inhibitory signals, shaping our behaviors and even our survival. Through Carl Jung's lens, we can see this as more than just biology. It's a clue to how our evolutionary past, our collective unconscious, and our individual psyche are all connected. This discovery is like a bridge between the hard science of genetics and the deeper questions of what it means to be human. So what have we learned today? A tiny genetic switch, mini exon B, plays a huge role in balancing our brain's excitatory and inhibitory signals, shaping our behaviors and even our survival. Through Carl Jung's lens, we can see this as more than just biology. It's a clue to how our evolutionary past, our collective unconscious, and our individual psyche are all connected. This discovery is like a bridge between the hard science of genetics and the deeper questions of what it means to be human. So, what have we learned today? A tiny genetic switch, mini exon B, plays a huge role in balancing our brain's excitatory and inhibitory signals, shaping our behaviors and even our survival. Through Carl Jung's lens, we can see this as more than just biology. It's a clue to how our evolutionary past, our collective unconscious, and our individual psyche are all connected. This discovery is like a bridge between the hard science of genetics and the deeper questions of what it means to be human. Thanks for joining me on this journey. If you loved diving into this topic, hit that like button, subscribe, and let me know in the comments. What do you think about Jung's ideas meeting modern neuroscience? Want more videos like this? Let's keep the conversation going. Until next time, keep exploring the mysteries of your mind.